Okay, a long time ago in a faraway land, speed this up, there was a group of students who learned about DNA structures. That's you guys. Okay, we're going to review the first few slides are just reviewing DNA structure. So what are the three parts of a nucleotide? Yes! Phosphate group? Nitrogen base? And the 5-carbon sugar. Very good. You guys can throw it out there. Here I have a picture on the screen of a nucleotide. Remember when we learned about the nucleotides, we learned that we must be able to count the carbons on the ribose sugar. Good. So we'll start with the carbon that's attached to the nitrogen base. That will be carbon number 1. And then we'll move away from the oxygen. You guys remember this? Okay. Let's see if I can. Carbons. Because of this, we know that DNA has direction. In the top of my picture, would this represent the 5' side or the 3' side of my nucleotide?
5'.
5'. And the bottom down here?
3'.
3' side. Very good. Becomes important today. What kind of, see if you guys can remember this, what kind of bond puts 2 nucleotides together here? Phosphodiester bond. And still got 5', 3', we got this down. The strands run anti-parallel, meaning one of them is the 5' side going down, and the other one is the 5' side going up. So we said the strands are parallel, and they are, but they run different directions. That's what the anti comes from. Questions on that? Still review? Okay. What kind of bonds are here in pink between the A and the T?
Hydrogen bonds.
Good. And how many hydrogen bonds are between A and T?
2.
How many hydrogen bonds are between C and G?
2 + 1.
Very good. Phosphodiester bond. Thank you, Kristen. Purines. Who are the purines?
A, G.
A and G. Remember, ag is pure.
And food.
Do purines have single ring structures or double ring structures?
Double.
Purines are double ring structures. If you look at the picture, A here has 2 rings, and G also has a double ring structure. Pyrimidines, T and C, are single ringed structures. Is it all coming back to you guys now? Yes? Okay.
It's like all
A always pairs with T, 2 hydrogen bonds. C always pairs with G, 3 hydrogen bonds. Moving on. Here is the first new part of today's material. Who remembers one major difference between prokaryotic and eukaryotic DNA? Histone proteins, okay? Remember on your semester exam, there was this question, and it was like, oh, the DNA in prokaryotes or the DNA in eukaryotes, which one is associated with proteins? Answer is eukaryotes, and they have histone proteins. So what we need to know about this is that there are 4 kinds of histone proteins in this core, but there are 2 of each kind in the core. So how many total proteins will be in this core?
8.
8. 4 of each kind, 4 kinds and 2 of each kind. And they make up one core. That core is what the DNA wraps around. So the DNA in this picture is this blue stuff.
What's the yellow stuff?
Not there yet. This blue stuff, the DNA, wrapped around one core has a name. Does anybody know what it's called? Starts with an N. May or may not have heard this word before. Nucleosomes.
Oh.
So nucleosome is DNA wrapped around the histone core, which has 8 proteins. And nucleosomes, IB says they want you guys to be able to state this. Like if they say some question on a short answer test, and they want you to state that nucleosomes help to supercoil DNA. So the idea that we have these cores here, which the DNA can wrap around, helps us to organize our DNA. So you should write down nucleosomes help to supercoil DNA. Like an old lady with curlers in her hair, right? You got to organize all that hair. You're going to wrap it around curlers. Now, there are actually more than just 4 kinds of histone proteins. There's other kinds, which you can see here in yellow, that are located not on exactly a part of the nucleosome core, but maybe on other places in the DNA that help to keep it organized. Those are additional histone proteins, not a part of the core. So what this would look like, let me see if I can. If you were looking at eukaryotic DNA, you'd have like a bead, then you'd have a line, then you'd have a bead, and a line, and a bead. Now this is with a really high-powered microscope, obviously. Right? It looked like this. How come you can't see that? There we go. So you can see this. These balls here would represent the nucleosomes, and then somewhere along these strands you might have other histone proteins with helping for the organization. This is all unwound. Now when we wind this up, then you have like, see that? What is it starting to look like? Right. So when we see chromosomes, they're usually already supercoiled. Something like that. I was trying to draw nucleosomes and then each one. Okay. Questions on packing of the DNA? Then let's move on to types of sequences. Now, your entire life, your science teachers told you that your DNA is responsible for making your proteins. DNA holds the instructions for making proteins. However, only about 2% of our chromosomes actually codes for proteins. The rest of it is other stuff. Most of it's useless, but we'll talk about that in a minute. So we have 4 kinds of sequences that you have to know. Each of these 4 I'm going to go through now. First one, highly repetitive sequences. There. Highly repetitive sequences. In human DNA, we have these sequences of base pairs that are just repeating units. Repeat, 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 repeat. Sometimes 5 to 300 base pairs. Now there's 2 possibilities here. They can be put in a discrete location, which I'm not really sure what that means, but the book said in a discrete location. Every book I read was like, in a discrete location. These repetitive sequences are called satellite DNA. But most of these repetitive sequences are just dispersed throughout our genome, right? Now, here's what's the weird thing about these repetitive sequences. They do not appear to have any function. So they don't code for any proteins. They don't actually do anything. However, what's really weird is that these genes can change places on a chromosome. So they're transposable, and they have like a nickname of jumping genes because they move from one place to another within a chromosome. Yes? Okay, so highly repetitive sequences. Number 2, protein coding sequences. These are the ones we are most familiar with as far as biology goes because we studied them 9th grade year. These are regions of DNA that code for a particular protein, that code for making hemoglobin, that code for making lactose, that code for making something in you that's a protein. Now, even within that gene, let's say you have a section of DNA and it codes for hemoglobin. Even within that section of DNA, there's sections that don't code for anything. You might remember this from 9th grade year. Do you remember that DNA, when we're doing transcription and translation, we have to cut out the introns and splice together the exons? Do you remember that? So even within that gene, we have regions that don't code for anything. But this is a good thing, because actually when we're transcribing genes, we can combine them in different ways to make a wider variety of proteins. Okay. Structural DNA. Structural DNA is highly coiled. Probably an area with a lot of what? Histone proteins, right? They have no coding function. They don't code for any proteins or anything like that. They're strictly structural. We find them around the centromere. What's the centromere? Right. Let's see if I can show you a picture. Do you remember this thing? It's weird that I'm drawing. You can't see it. Ha ha ha. Ta-da! Do you remember this red part in the middle? Oh, messed up my arrow. Ah. Okay, the centromere, right around the centromere, areas in here, oh, it's like, whatever. Areas around the centromere have structural DNA. We don't want our genes too close to the centromere because they could get damaged. And that would be bad. Questions?
How can genes get damaged when they cross the centromere?
Well, the centromere is a point for holding them together. And we have to pull this apart during replication. And so it's kind of like being really close to the edge of something.
Okay.
If it breaks or anything else bad happens to it, get damaged. Now, short tandem repeats. This, yes.
So you're saying that structural DNA is basically like a buffer between...
Right. And actually, I'm going to give you another example when we talk about replication at the end of this lecture. And you're going to be like, wow, it's a really good thing we have that. But I don't want to give it away yet. Okay. Short tandem repeats are polymorphisms. If you sat here and you thought about it for a minute, right? Did anybody ever hear that we're like 97-ish% identical to a chimpanzee? Right? Our DNA. And, oh wow, that's pretty similar. But do you know how similar we are to each other? Some really, really high percentage, like 99 point something. So the reason behind that is that Rachel and I actually make almost all the same proteins. Rachel, she makes hemoglobin, I make hemoglobin, right? She makes collagen, I make collagen. We make the same kind of proteins. Now we have a few small differences because somehow her DNA produced this really beautiful young lady, and I'm just like this old lady, right? So our proteins are slightly different, but that difference is so small that actually our DNA is almost identical, 99-something identical, right, between me and you. So how is it when we do DNA profiling that we can tell the difference between the suspect and the crime scene?
Specific regions have variation.
Yes. When we do DNA profiling, that very, very small difference is found in this area, polymorphism, right? So you guys, we're going to actually do this experiment. But you know you've seen the gel, and it has like suspect and crime scene. And then you have like DNA bands. You've seen this?
Yeah.
And then the crime scene is like, oh, like here, and it's like, oh, well, actually, that doesn't really match. So that guy didn't do it. Last block, he did it. Those sections of DNA that are actually different are called polymorphisms or short tandem repeats. And IB wants you to know that we use short tandem repeats for DNA profiling. Short tandem repeats can be used for DNA profiling. Oh, did I go too fast? We're good?
It's the mesyl.
Mesyls and stalls. So here's what we need to talk about. That's enough about sequences. Let's talk about how we copy these sequences. Another experiment. This unit seems to have a lot of these kind of things in it, right? Experiments. These two guys wanted to find out how DNA is copied. And there's three theories. Before they did this experiment, there were 3 ideas out there, and they look like this. Let's see if I can draw it for you. Okay, this is your original strand. Oh, nice. Okay. And when it's copied, one copy is all the original strand, like this. And one copy is all new DNA.
Is that what they thought?
This is one of the theories. So this theory is called conservative replication. Okay? We're completely conserving the old strand, and we're like basically photocopying to make a new strand of DNA or a new double helix. Conservative. And then there was this idea that maybe only one side is kept in each new strand, and the other side is new. So it looks like this. Does that make sense? This method is called semi-conservative, meaning one old strand and one new strand is on my new DNA. Then there was one last theory, which is called dispersive. And dispersive says that chunks of DNA are copied together. So yes, the new DNA has some old and some new, but in chunks like this.
What's that called?
Dispersive. Which one do you guys think is actually the way it happens? Semi-conservative, because now we know. But we didn't know at the time, and so this is the experiment they did. They took this N15. So this 15 means that they grew nitrogen with a heavier isotope. So it's literally a heavier nitrogen than a regular nitrogen, okay? And they took this bacteria and they grew it on just heavy nitrogen. So the only nitrogen available to this bacteria is N15. So when the bacteria make all their nitrogen bases, they use the heavy nitrogen. So they took this colony that they grew and they put it in a centrifuge, right? And you can see here, all of the DNA is very dense because all of the DNA is now made with this N15. Got it? Okay. Now, all we have to do is now take that first colony and put it on regular nitrogen broth and let it have one generation. This is tricky because this DNA, let me see if I can clear this picture.
You watched the video?
Huh?
The video?
No, I'm not going to show you the video. Okay, so if, I can see if I can do this here. If they started out and this was the original, right? This DNA is heavy. After the first replication, we now know that one of these strands will be heavy, and what will the other side be like?
Lighter.
It'll be the lighter. So the DNA is 50% as dense as it was before. Does that make sense to you guys? And then same thing on the other side. But if they replicate again, it's going to be cut in half again, because actually I still only have my 2 original strands that are heavy, and the rest of them are all new DNA with the lighter nitrogen. Does that make sense to you guys? So a good IB question or a good test question would be to show you this picture and ask you to explain why the amount of DNA that's actually still heavy is decreasing. As each replication happens, all of the new DNA is made from the new N14, and only the original 2 strands contain the N15. But these percentages helped us see that actually DNA is semi-conservative. So let's take a look at how this happens. Skip this one, semi-conservative. Replication. Why would I want to do DNA replication? For what purpose?
To grow.
To make more what? Not proteins. For what reason would I need more DNA?
Cell division.
Cell division, right? I need to make more DNA. All of my chromosomes have to be copied so that I can divide the cell. So step number 1 involves unwinding the DNA. What bonds are broken here? Hydrogen bonds. And does anybody know the enzyme? Helicase. Then we have these light blue things, these light blue circles. Those are single-stranded binding proteins. They actually hold the double helix open. Question? Yeah.
Do the single-stranded binding proteins just like move up the base on the do like stay?
That's a good question. I'm not sure. The question is if they move with the helicase. I would imagine that after the replication happens, they let go and new proteins bind. Question? Idea?
I think they were doing, like, they made videos of this or something. Yeah.
Want to share?
It was an extra credit image.
And what was it?
Like they videotape the process of DNA replication, and then whoever liked it, whoever got the most likes or something would get extra credit.
Nice. Okay, so there's another enzyme that IB didn't say you need to know, but I'm going to tell you because, of course, you all want to get a 5 on your AP exam, and should it show up, I wouldn't want that to be a deal breaker. So there's this other enzyme that's found here just down from your replication fork called topoisomerase. Topoisomerase. It's spelled just like it sounds. TOPO isomerase. And it helps release the tension as the helix is unwinding. Because you can imagine, like, if you have this double helix and you pull part of it open, it's going to create tension on the strand down from where you pulled it open at. So topoisomerase helps to relieve that. And on a single DNA strand, you actually have these bubbles all over the place. It's not one bubble of replication. You'll have a bubble here, a bubble down here, a bubble down here, and they'll be going in both directions so we can replicate our entire genome really fast.
What happens when both helicases meet?
The DNA will be completely separated.
Oh.
And they the enzyme that adds the new nucleotides can put 4,000 bases a second. That's how fast it works. 4,000 bases per second. Crazy, huh? All right, so this says replication second step. It's actually sort of incorrect. The second step for replication is the adding of an RNA primer. So for this, you can just make a little correction on your notes or whatever. RNA primer has to be added before we can replicate the DNA. It talks about it a little later in the PowerPoint, but it seems out of place to me. So RNA primer is laid by an enzyme called primase. And primase puts down some RNA nucleotides. Now that happens so that DNA polymerase 3 can come along and start to copy the strand. So let's talk about what that looks like. Have you guys seen this before? What is that? ATP. Turns out ATP is a nucleotide with 3 phosphates. It has another name, but it's not the only one. There's GTP, TTP, and CTP. And all of those guys come with their own energy because we're building DNA, right? And building is what kind of reaction? Anabolic. So we need to put energy into the system. These molecules have to come with their own energy. There we are. There they are. Let's see what this looks like. Nucleotide, you see the 3 phosphates? Phosphates break off. DNA made. Okay, another one on. Phosphates break off. DNA made. Keep going. No problem. And wow, look, I built a DNA strand. Yay! Okay.
Is this where we get our ADP from? No, JK.
All right, but there's a problem. You guys already know what this problem is. You probably remember, but let me show you. Let's say this is the DNA strand I want to copy. So I split it down the middle. What bonds am I breaking? Hydrogen bonds. Very good. Now I'm going to copy my right side first. And if I'm copying the right side, my new strand is got to form from the 5 prime, right? So 5 prime, then phosphates break off, right? No problem, right guys? Okay. Now, on the other side though, it's the other side. It's the 3 prime side that I'm going to be making right here. And I'm going to have a problem, and here's why. Here's first base, no problem. Second base, there's a problem. What's the problem?
They break off.
The phosphates are not in the right place to form a bond here. You see that? The phosphates have to be wherever they're making the bond at. So phosphates break off, no bond formed.
Wait, why?
It's facing the wrong direction. Yes, yes. So this doesn't work, right? So I have to start from down here and go up. Now it works. No problem. No problem. So you can only build your new strand from a 5 prime to a 3 prime. Same thing over here. From 5 prime to 3 prime. That's an important fact you have to know. DNA polymerase 3 can only build your new strand from 5 prime to 3 prime. We just saw it does not work the other direction. All right, this side makes sense, right? You see when I set this nitrogen base here, the phosphates overlap where they're going to bond at. Okay, orientation. So that's fine. They'll create the bond. On the other side, when I go to bond the 2 nitrogen bases together on the same side, the phosphates don't face each other. Right?
But why does that matter?
That energy is the energy that creates the bond. So unless I use some ATP or something here, this bond will never form. So I've got to form it from the 5' to the 3' side, and then it's not a problem. But if I continue to do that, at some point I've got a hole. You guys see the hole? Whoops. See the hole right here? Okay. So we have to have an enzyme that comes back in and seals that hole up. Does anybody know what that enzyme is called? Ligase. Ligase will seal that up. We'll talk about it more in a second here. Alright, so because DNA polymerase III can only build a new strand in a 5' to 3' direction,
oh, then you have the lagging.
You have a lagging strand. You have a strand you have to build backwards. We just saw that. So what's this yellow thing here represented? What is this yellow thing? Helicase, right? There's helicase. All right. Let me put my 5' and 3' up here. Oh, what are these things in black? Single-stranded binding proteins. Good. This is my replication fork. It's going that way to the left. The top is my 3' and the bottom is my 5'. So down here we have 5' and 3'. Everybody's good? Okay. Based on this information, which strand is my leading strand? The top or the bottom?
Wait, isn't top
The
lagging?
bottom.
Top is lagging. 5 to 3 would be the.
So the bottom here has 3' side, right? Which means my new strand will be 5'. And that's the direction I need to go. This one up here is the 3'. Here's my DNA polymerase III. Before that can attach, what do I need? RNA primer, which we'll get to in a minute. It's not shown in this picture, but it has to be there. Okay, and then here, 3' to 5' won't work. But if I start on that side and move to the right, it works. But then I get these fragments. They're called Okazaki fragments. And I can use ligase to seal them together. My leading strand is on bottom, my lagging strand is on top. A great quiz question would be to show you this picture and ask you, without any of the other stuff, just the original DNA, which is my leading and lagging strand. So let's take another look at it. It depends. In a bubble, we're going to go both directions. Actually, I'm going to show that to you now. So let's say this is my DNA this time. And I've got a bubble, and I'm going to actually do replication in both ways from my bubble. Okay?
And then would you have a different leading and lagging strand?
You do, and I'm going to show that to you. So look at here. If I'm going to the left, which one is my leading strand? The top or the bottom?
The bottom.
The bottom. But if I'm going to the right, which one is my leading strand?
The top.
The top. Do you guys see that? Going to the right, that works. 5' to 3', yes. Going to the left is going to be a different story. So actually your leading and your lagging strand changes depending on which direction you're going. If I had labeled these differently, if I made this my 5' and this my 3',
You reverse.
that would totally change the picture. So you have to pay attention to how it's labeled. Alright, RNA primers. So what is the RNA primer needed for?
Priming the DNA.
DNA polymerase III to attach. We need RNA primer in order for DNA polymerase III to attach. So everywhere you see the light blue on this picture, starting, we had to have RNA primer first. So what looks like this, RNA primase lays an RNA primer, and then DNA polymerase can attach. RNA primase lays an RNA primer, and then DNA polymerase can attach.
Does RNA have to work in the same Okazaki fragments as?
Yes.
Okay.
So each time we want DNA polymerase 3 to attach, we lay down the primer first, and it works. What's the problem with this?
Yeah.
Now I have a bunch of what in my DNA?
RNA.
I have a bunch of RNA in my DNA, right? So now I got to get rid of that RNA.
DNA polymerase 1.
DNA polymerase 1 is going to get rid of my RNA primer. So the sequence of events goes like this. Actually, we'll get to that in just a second. I'll recap it for you. There it goes, all my RNA primer. However, we have a problem down here. We have a problem right here. We cannot attach DNA polymerase 1 here and work backwards. It has to work in the same direction. It can't attach to nothing and work in the same direction. So every time we do DNA replication, there's some part of that DNA strand on the original leading strand that gets shorter.
Is that why we get older? And then that's mutation? No, I'm stupid.
Actually, it's true.
What?
So the ends of our DNA have something called telomeres. And telomeres are a type of structural DNA. Remember we talked about structural DNA? Telomeres get shorter every time we replicate. If this was a gene, we'd have a serious problem. But since it's on a telomere, which is a non-coding region, just a structural thing, it's okay. The longer your telomeres are, theoretically, the longer you could live for.
So,
Or if we made people genetically having longer telomeres. Much longer. If no other factor played a role in your death, eventually you would die because your telomeres would be gone and you would die of old age.
but if you find a way to replace it every time, then would that be immortality?
It's a possibility.
Why wouldn't you want to live forever?
Is the world freaking awful?
Heaven's calling my name. Okay. DNA polymerase 1, we got a problem there at the end. Good. Just talked about that. Loss of bases at the 5' ends in every replication. Is there a limit to the number of cell divisions? Some people believe, and I'm not sure how much scientific basis is behind this, but it's not real. It's not a far out there thing. Okay, this might be a true thing. If you ate less food, if your body was on the verge of starvation, your cells actually divide slower. And you could theoretically live a longer life.
If you starve yourself.
Not starve to death, but yeah, keep your body like just under the amount of calories you needed to slow down your cell division.
Interesting.
Okay. And I believe we just went over this. Okay, so here's the whole recap. A replication fork is where helicase begins to unwind some DNA. The DNA is held open by single-stranded binding proteins. RNA primer is laid by an enzyme called RNA primase. RNA primer allows DNA polymerase 3 to attach. DNA polymerase 3 can only build a new strand in a 5' to 3' direction. So on one strand, it is building continuously. This strand is known as the leading strand. On one strand, it is building discontinuously. This strand is known as the lagging strand. Everywhere the discontinuous DNA is laid, they must be sealed together. This is done by the enzyme ligase. DNA polymerase I can then come in and replace RNA primer with DNA nucleotides, eventually leading to 2 new DNA strands. DNA polymerase III?
Yeah.
Nope, it's the same. You can't tell after the DNA's been replicated where it was that happened. Question?
What happened to DNA polymerase
DNA polymerase II, that's a funny thing actually. There are about 8 DNA polymerases in eukaryotes, and you only have to know 1 and 3. But there are 2, then there's other polymerases also. We talked about how fast it can go. Keep going, keep going, keep going. We try not to make any mistakes. 1 E. coli can copy 5 million base pairs in about 30 minutes. A human cell can copy 6 billion bases in just a few hours. Your cell can divide.
If I had $1 for every single cell base that I copy, I'd be rich.
Yes. And there's only 1 error per 100 million bases. So it's really